will defend the academic thesis Modifiable Risk Factors of Non-Communicable Diseases, Epidemiological Approaches and Methodological Considerations. Welcome, dear candidate.
Thank you so much.
I would like to invite you to present a summary of your study and the conclusions of your thesis in a presentation.
Thank you so much. I will now share my screen. Yeah. So, dear Pro-Rector, dear members of the assessment committee, dear supervisors, dear colleagues and friends, thank you for being here today. I will now present a summary of my PhD dissertation titled Modifiable Risk Factors of Non-Communicable Diseases, Epidemiological Approaches, and Methodological Consideration. The dissertation focused on two of the major cancers, bladder cancer, which is the 9th most common cancer, with more than 600,000 new cases and 220,000 deaths annually, and prostate cancer, which is the 2nd most common cancer in men, with 1.5 million new cases and 400,000 deaths annually. So with this major burden of the diseases, non-communicable diseases, and specifically cancers, the prevention of non-communicable diseases requires identifying modifiable risk factors, such as the nutritional factors, because diet is modifiable, and also everyone is exposed to dietary factors. The problem is that nutritional factors is difficult to study because nutrition is too complex. And this complexity may partially come from the various stages that food goes through from production until consumption by humans. So at the first production of the food, which can be exposed to pesticides, soil contamination, antibiotics in animal production, for example, and then go through processing, where preservatives, added sugars, trans fats can be added, in preparation with various methods of preparation of foods, and some of these methods can create some harmful substances in the food. And then the composition of the food itself, which is each food is composed of multiple and too many nutrients. Some foods can be stored, which lead to degradation or leaching of chemicals and mold formation. All of these, after consumption of the food, can lead to changes at the cellular and tissue level that can progress to cancer. And to add to all of this complexity, other factors like genetics, lifestyle, microbiomes, medications can interact with the exposure of a human to these toxicants, and also interact with the progression of the cellular changes to the full-blown cancer. So nutritional epidemiologists try to figure out the association between various nutritional factors and non-communicable diseases, and three main approaches have been used in nutritional epidemiology: ecological studies, observational studies, including cohort and case control, and experimental studies. Of course, each type of the studies have its own pros and cons. So for ecological studies, for example, it provides a large contrast in diet, which is not usually the case in observational studies, and a stability of national diet compared to individual variation in the dietary. But also has its own disadvantage, like being susceptible to confounding and ecological fallacy, and its dependence on food disappearance data to measure the exposure to diet. Observational studies, on the other hand, provide the ability to study individual level data in their own real world, but also are susceptible to other negatives or disadvantage, like being susceptible to confounding and sort of bias. The experimental studies, which is usually considered the gold standard for assessing causal effects in nutrition and epidemiology in general, however, it has its own problems, because there is a problem of the weak generalizability of the experimental studies because it usually focus on selected people. And then other difficulty like blinding and long-term adherence of participants and the post and sample size. So most of the nutritional epidemiology used the observational studies to study the nutritional effect. So with nutritional observational studies having some contradictory results, we need some evidence synthesis beyond the single studies. So we need to collective evidence that synthesize the results from various observational studies into an aggregate estimate. So combining the smaller data set, which amplifies the statistical power. And three main approaches in evidence synthesis include the meta-analysis, pooled analysis, and overview review. So meta-analysis, we are combining the results, the overall results of multiple original studies, which can enhance statistical power and precision. In the individual participant data meta-analysis, we are combining raw participant level data, which allow us to analyze individual level covariance and investigate interaction and rare exposures. In contrast, the overview of reviews allow us to compile evidence from multiple systematic reviews and meta-analysis, which is an efficient summary of risk factors and can highlight the knowledge gaps. And these approaches have been implemented in Chapter 4, Chapter 5, and Chapter 2 and 3 in this thesis. Another approach that is recently becoming more and more commonly used in nutritional epidemiology is the machine learning, which provide advanced analytical techniques for predictive modeling, allow us to handle complex multidimensional data, which is common in nutritional epidemiology, and handles nonlinear relationship and interaction, integrate diverse health data from different sources, and also potential for tailoring risk assessment for developing prognostic model, which this approach was applied in Chapter 6 in the thesis. So our thesis in Chapter 2, we studied modifiable risk factors for bladder cancer and prostate cancer, respectively. And then in Chapter 4, soft drink consumption association with asthma, tea consumption in Chapter 5, association with bladder cancer. And in Chapter 6, we utilize the machine learning to study nutrient as a predictor of bladder cancer. So in Chapter 2, where we studied the modifiable risk factor for the prevention of bladder cancer, the aim was to provide a comprehensive overview of these modifiable risk factors, utilizing systematic review of published meta-analysis, searching various databases, including 82 meta-analysis, and conducting meta-analysis where appropriate, and also calculating the probability of causation for significant factors. So the overall 19 lifestyle factors and 48 occupational factors were found to be significantly associated with bladder cancer. For example, fruit intake, vegetable intake, physical activity, farmers' occupation, and gardeners all have reduced risk of bladder cancer, while current smoking, of course, and also processed meat intake, tobacco workers, dye workers, chimney sweeps have increased risk for bladder cancer. In Chapter 3, which is also have utilized the overview of reviews of meta-analysis, trying to summarize modifiable lifestyle, environmental, and occupational factors associated with prostate cancer, searching various databases again, and including 260 unique risk factors, and conducted meta-analysis where appropriate with assessment of risk of bias. Overall, 26 dietary factors, 13 lifestyle factors, and 22 occupational factors were found to be significantly associated with the development of prostate cancer. For example, calcium supplements and olive oil were found to be associated with lower risk of prostate cancer, and while fried foods and dairy products found to be to have increased risk for prostate cancer. Again, also sunlight exposure later age at first intercourse, decreased risk, while lower education and more sexual partners have increased risk. In chapter 4, we move to the association of soft drink consumption and asthma, utilizing this time a systematic review and meta-analysis of original primary studies, searching various databases to find out the primary studies to be included, and overall that include 19 studies were found to study this association with about 500,000 participants and 50,000 asthma cases. We conducted meta-analysis using random effects model with quality assessment of primary original studies. And the findings showed the adults have increased odds of prevalence of asthma associated with soft drink consumption, and also a prenatal exposure of pregnant women increase the risk of their infant to development of asthma. In children, we are able to estimate the overall, which is an increased risk also, and also subgroup analysis for sugar sweetened soft drink and carbonated drinks. We found that there is an increased odds of sugar sweetened soft drinks, but not in carbonated drinks. And various mechanisms have been proposed in the to explain the association between soft drink and asthma, like sugar content activating inflammatory pathways, or fructose specifically leading to increased lipogenesis and inflammation. The additives added to these soft drinks can play a role, or it could be indirect effect through soft drink leading to overweight, obesity, which can lead to exacerbation of asthma. In chapter 5, we studied the tea consumption and risk of bladder cancer using a pooled analysis of 12 international cohort studies with around 500,000 participants, utilizing the blend consortium dataset and using Cox regression and dose response analysis. The main findings showed that tea consumption is associated with bladder cancer across all the levels of the exposure, and with a dose response relationship between the tea consumption and the bladder cancer. So stratified analysis shows no significant association in females and never smokers. Again, here, potential mechanism of the association between tea and bladder cancer could be related to the tea components, which is usually suggested as a cause, mainly polyphenols, especially catechins, which may inhibit cell proliferation and suppress angiogenesis or induce apoptosis in cancer cells. And possibly there was some interaction with the smoking-related carcinogenesis, as is shown in the results of this study. In chapter 6, we move to studying nutrients and risk of bladder cancer using a machine learning approach to identify nutritional predictors. Again, using the blend data set, pooled analysis of 14 international cohort studies, and utilizing various algorithms in machine learning, like decision tree, logistic regression, random forest, neural network, and naive Bayes. And these models have shown good prediction of the outcome across the various algorithms, as shown here. And then when we focus on the important factor or risk factor of both nutrients, and specifically in logistic regression model, it shows, for example, thiamine, phosphorus, iron, and cholesterol as the most important features or risk factors that have been included in the machine learning algorithm. So take-home message that modifying lifestyle and occupational risk can greatly reduce bladder and prostate cancers. Soft drinks are a threat to your breath, so with a study showing association between soft drinks and asthma. Some tea to help reduce your risk of bladder cancer with as a result of the study and the association between tea and bladder cancer. And then from methodological perspective, establishing standards is the key to avoid a methodological miss in nutritional meta reviews, which is a new approach where a lot of standards have been established. Combining individual data strengthens nutrition cancer investigations and overcome the problem of small sample size. The future is now advanced analytics and big data are the new allies against non-communicable diseases. That's all, and thank you so much.
Thank you very much, Mr. Alzalabani, for this very clear presentation and interesting findings. We will now open the opposition, and the first opponent will be Professor Smits. He's a professor of clinical epidemiology and risk-based care at Capri Institute of this university in Maastricht. So the floor is yours, Professor Smits.
Thank you, Pro-Rector. Dear candidate, congratulations on the publication of your thesis. I've never before read a thesis that fully consisted of meta-analyses, so I was a bit worried about the variety of studies that you were presenting to us, but well, on a closer look, your thesis showed a variety of study designs and goals, so that was fortunate. And I really have to compliment you on the comprehensiveness of that you demonstrate in all these studies, and also on the presentation that you just gave to us, which was very nice. Still, I have some topics that I would like to discuss with you. They all concern chapter 6. And in that chapter, you use several techniques to identify nutritional factors, nutritional predictors of bladder cancer. And you also compare these techniques. And logistic regression was one of them. And I'm a little bit an old-fashioned researcher. I like the feature of logistic regression a lot, that it gives association measures for each outcome. And with that and some extra information, you can calculate what proportion of the disease you might prevent when you reduce the exposure to that factor. And that is, of course, very important for prevention workers. So, and as far as I know, the other techniques do not provide such association measures. So my question is, that's my first question, wouldn't that be a huge problem if you want to use these techniques when prevention is your goal?
Thank you so much, highly esteemed opponent. Of course, the machine learning approaches and machine learning algorithm has its own advantage and also their own disadvantage. Similar advantage of machine learning, so allowing, for example, the high dimensionality in nutritional epidemiology. So, for example, here we have nutrients. We have a lot of nutrients to be studied at the same time, and also dealing with, for example, if in other settings where we have various sources of data that we can know, and other also advantage of the various machine learning algorithm that can allow us. However, definitely the machine learning is mainly concerned, most of the algorithm will give us a prediction of the outcome rather than detailing the association and the risk or preventable risk of the outcome of the diseases. So it's mainly as a predictive modeling to predict the outcome, and during or utilizing this prediction, we can identify factors that can be a potential risk factors for the outcome or the disease, and then we can conduct more studies, or traditional epidemiology studies that allow us to get the estimates of the preventable.
Okay, yeah, I already expected an answer like that. So you are arguing that you can use these other machine learning techniques more to identify the factors instead of estimating the strength of the association. But then again, how can you tell which one of these techniques does the best job? Because you use all kinds of discrimination measures. I understand that, and not calibration, because discrimination is more dependent on the strength of risk factors than calibration. But they only say something about the predictive performance of the model as a whole, but not about its capability to identify individual risk factors. Or do I get it wrong?
Thank you. I would say that if in a setting where we are concerned or we are interested more in the prediction of the outcome, so we can use definitely machine learning and to predict the outcome if we have some risk factors that can be easily measured in the clinical setting, for example, or in the community setting, and then we can predict the outcome. And so here will come at our surface the machine learning algorithm that can help us a lot. If we are more interested in the association and the risk factors, now we have the overall estimation of the prediction or predictive power or discrimination power of the machine learning algorithm. And then we have the feature importance methods, like what we have utilized in the thesis, which is permutation feature importance, that can indicate which factors or which features were more important in the prediction. It doesn't give like a risk ratio or a noise ratio or an estimate, but it gives the ranking the importance of the features, and then we can proceed to studying or utilizing other method to get the estimates or the risk estimates.
Yeah, I know what you're referring to. For instance, the graphs on page 272. And there you show all the risk factors that were most important in all of these techniques identified by all these techniques. And in the conclusion section, you say that the factors that were identified by these models remained robust across the different modeling algorithms. But I doubt whether that is really true. Because you have 5 modeling techniques, but only one factor, cholesterol, was identified by more than 2 techniques. So, and all other factors by only 1 or 2 of the 5 techniques. So, how can we tell which technique identifies the right factors? So, and which doesn't? We don't have a gold standard, of course.
Thank you. Yeah, so in the figure in page 272, we presented the most 5 important features in each algorithm. So we, for the sake of the space, we didn't present the whole, like for example, the most 10 important risk factors or the whole risk factors ranked in the importance. So although in the most 5 important, cholesterol was the common factor, but if you go through more risk factors or more features, we can find that these factors are still consistent among the algorithm, although the ranking is different between them. So I agree that the prediction or the prediction models or machine learning algorithm probably will not help in the assessing the absolute risk or the relative risk of specifically for a disease. However, it can identify the most important, and then utilizing other approaches, we can specify the degree of the association.
Okay, okay, I understand what you are saying, and for now I'm content with your answers, and I would like to give the word back to the proactor. Thank you.
Thank you very much, Professor Smits. We continue the opposition with Professor Jonkers. She is the Scientific Director of NUTIM Institute and Professor of Intestinal Health. The floor is yours, Professor Jonkers.
Thank you very much, dear candidate. I also would like to congratulate you and your supervisory team with a very interesting thesis. I'm very much interested myself in diet in relation to inflammatory diseases, intestinal diseases like inflammatory bowel disease. So for me, it was really interesting to learn from your approaches to actually go beyond the single studies. You use the collective evidence, but also to add new analytical approaches. So very interesting. And you started off with a very nice presentation mentioning the complexity of diet, which I totally agree, which is kind of a challenge. And then I came across chapter 5, and then you kind of simplified diet by focusing on tea. Maybe first a very simple question: why did you decide just to focus on tea in chapter 5?
Highly esteemed opponent, thank you so much for your compliments and comments. In chapter 5, we have studied the association between tea consumption and the bladder cancer, and this is part of the a bigger project of the blend study, where various factors, nutritional factors, have been investigated in the to find out the association. So tea is very ubiquitous. I mean, a lot of people around the world is exposed to tea. So if you find even the slightest effect of the tea consumption, that can translate to high or large population impact. So this is one reason of going to yeah.
Yeah, I even saw a proposition that a sip of tea may help to reduce the risk of bladder cancer. So I'm really intrigued to understand how that works. And you already mentioned that you consider the polyphenols, that's what you mentioned in your presentation, as a key factor there. Can you comment briefly on that? Do you think that's the main factor in tea which is responsible for the reduced risk you see?
Yeah, so polyphenols, specifically catechins, play a role and found in various studies, actually in vitro, in animal studies, and in the human studies, that the polyphenols have an antioxidant and anti-cancer effect. So that is probably the most important candidate of or component of the tea that probably have played a role in the prevention of cancer. Also, other theories or hypotheses have been proposed also in the effect of tea and association with the bladder cancer, interaction with smoking, for example. So in our study, the effect was prominent among the smokers group compared to never smokers. Which leads some others to state that probably the effect of tea is counteracting the toxic effect of smoking.
I was intrigued by that indeed, because you know, I'm not an epidemiologist, I'm a nutritionist. I try to understand, you know, what's happening. So then I saw indeed that it was protective, but I thought if it is lowering the risk of smokers, then it may reduce the relative risk of smoking. But why it is then really ending up as a protective factor?
Yeah. So
is it just the smoking effect, or is it broader?
Yeah, that was also debated in the literature, that the effect is mainly reducing the smoking effect, which is again has its merits, this point. However, if we look at the results, we can see that the effect is even consistent. I mean, yeah, it is consistent among all groups, but it's more prominent among the groups of smokers. So this can lead us to conclude that the tea is still a protective effect, has a protective effect for the overall population. However, probably for smokers is having more prominent effect.
I still can talk about this for a longer time, but I'm going to switch a little bit, because the polyphenols, you triggered my interest with that, because they are also present in like fruits and vegetables, and for example, even in coffee. So the people who drink less tea maybe drink more coffee. So, you know, that actually brings me also to, isn't it not, you know, a bit of an oversimplification here to focus on the tea? And indeed, should we better go to the complexity of diet, which you indeed addressed in chapter 6, which I found very interesting. And actually, my first question was to follow up a little bit on the previous discussion and the features. And you showed the top 5 features, but I was a little bit involved in machine learning for other purposes, and I know that there's also a set that was then found to discriminate between, you know, cases and non-cases or whatever. So I was actually interested just to hear what was actually the number of features that you did find to find a discriminative effect.
Well, so we have estimated the overall performance, discriminative performance of the prediction model, our various machine learning algorithm, and compared these features using the performance measures presented, including AUC and other discriminative measures. So it gives us the overall discriminative power of the model. But then we can rank the features included in the model by importance. So it can be ranked along along the way. And then we can grasp the most important features of these factors, because rather than...
If I'm allowed to ask a last question, I look to the product. Oh, yes. So, because I'm very interested in this complexity of diet, you know, because it's so interesting, diet. I think there's something in that which relates to disease, but how to get it out. You know, here you focus on the nutrients. I always a little bit puzzled. Do we have to focus on the nutrients? Do we have to focus on the food items? Should we look to a more, you know, kind of overall quality score? How can you help me out? How to move further with the research in my IBD patients? How can I better get an insight in the complexity of diet, which really links to disease, like, for example, inflammation or cancer?
Thank you so much. Yeah, one point just to mention that in the study, even in the tea, we also having pooled analysis allowed us to adjust for other confounding factors, including, for example, the coffee and other nutrition. So for whether to focus on the nutrients or the foods or the diet, and I guess each approach has its own advantage and disadvantage again, because the nutrients are the most detailed level, but the problem is that each food is actually having multiple nutrients, and probably we cannot tease out which the effect of which nutrients. However, the nutrients has the advantage that if we identify one nutrients, we can provide a supplement, for example, for people to consume. So it has its own advantage also. Food, also another one. Diet, which is now nutritional epidemiology, I guess, is more toward studying the overall diet rather than...
I'm afraid I have to stop here. I would be very interested to follow up on you in a later phase on this complexity and how to catch that. Thank you very much for your answers.
Thank you so much, highly esteemed.
Thank you, Professor Jonkers. This is indeed a topic that you could spend another hour on, but we have to continue, and I'll gladly give the floor to Professor Salim. He's a professor of public health and community medicine at Maastricht University in Egypt. The floor is yours, Professor Salim.
Thank you. First, I would like to express my heartfelt thanks to Dr. Abdulmohsen and the supervisor team, okay, for this significant contribution in the field of cancer, non-communicable disease prevention. I have to go to chapter 2 regarding the probability of causation. Is it calculated directly by a simple basic statistical program, or calculated through the machine learning, or under the NIOSH program for Department of Labor, as it is originally developed for the relation between the cancer and radiation? This first question, please.
Dear highly esteemed opponent, thank you so much for your compliment and for comments. Regarding the question of about chapter 2 and probability calculation of probability of causation, in chapter 2, actually we have used the traditional epidemiology approach. We didn't use the machine learning algorithm, and probability of causation was calculated using the traditional software Stata software to calculate the probability of causation of each factors.
Regarding the combined probability of causation, it should be include protective and risk. Okay, because for 4 factors independent. Okay, and
could you
2
repeat the question,
factors.
Professor Salim? You were breaking up. Could you please repeat the question to the candidate? Can you still hear us, Professor Salim?
Yes.
Yes, you were breaking up. So can you repeat your question to the candidate?
My question about the combined probability of causation, it should be calculated, included both protective and risk factors, or no?
Thank you so much, highly esteemed opponent. So probability of causation, we tried to calculate the combined probability of causation for these factors without going into double counting, or I would say including factors that are also includes other factors. So for example, in vegetables and fruits, we have various types of fruits and vegetables, but we are trying to avoid that. So this is why we selected the 4 factors to be included in the calculation of the probability of causation. And we aim to have an overall estimate of the probability of causation, whether for the preventive factors or the harmful factors together.
Another comment regarding chapter 3, regarding the supplementary files 1 to 8 from pages 114 up to 214. Can summarization or highlighting the important or crucial data will be easy for the reviewer and for further study by other colleagues? Can this be helpful and visible?
Thank you so much, highly esteemed opponent. In the supplementary tables for chapter 3, we included various extra information that cannot be accommodated in the main article. So we included the RESMA checklist, of course, for reporting, but also included the best estimates the meta-analysis result, that for factors that we were able to calculate the meta-analysis according to the rules we defined earlier. And then we also listed the reasons for exclusion for cohort, case control, and randomized controlled trial meta-analysis, as well as the statistically significant factor and robust risk of bias result. So I guess all of these tables provide rich information for future researchers to utilize the information here, for example, in identifying the gaps, identifying the weaknesses or the problems that have been identified in a previous meta-analysis to be avoided in a future research. And also to fill these gaps with either to satisfy the proper methodology or satisfy the proper exposure or outcome measurement.
Regarding the machine learning models and utilization for future by other candidates, I think one important point to be highlighted in this respectable thesis is to suggest and recommend an obligatory advanced statistical course, including machine learning for postgraduate students in the future, to ensure their capabilities and ability to interpret the statistical results. Can simply explain what the basics of this could be helpful for the newcomers. Okay.
Thank you so much, highly esteemed opponent. Regarding this point, I think that as I have mentioned in the last slide, that the future here, because the machine learning approaches that is recently developed, combined with the richness or availability of rich data all over the world, utilizing the internet connection, utilizing the computer, utilizing the wearable devices that provide us, and in addition to electronic health record that becomes the standard in most of the healthcare institutions. So combining all of these data and utilizing the advances in the analytic and machine learning analytics is providing a strength point for any researchers, whether in nutritional epidemiology or in other discipline. So I guess preparing the future researchers in various disciplines to be able to deal with the big data, how to collect, what are the advantage and disadvantage of the big data, and how to utilize these data with keeping in mind the limitations inherent in the data, but also having the ability of analyzing and utilizing the advanced analytical techniques to do the analysis. I guess this definitely is an important competency that need to be included in various training program, especially at the postgrad training programs.
Thank you very much. And thank you, Professor Salim. We will now continue with Dr. Bossetti. She's a scientific project officer, of the JRC Institute of the European Commission in Italy. Floor is yours.
Thank you. Thank you very much, Prorector. Thank you, dear candidate. So first, congratulations for all the work done for your PhD and for your thesis. So you analyzed mainly risk factors related to nutrition and their association with some non-communicable diseases using various approaches. So you did a huge work combining many studies within review, systematic review, meta-analysis, review of meta-analysis. My question is focused mainly on chapter 6, which is the most innovative, and you already received some questions. So among the methods used in machine learning, you mentioned logistic regression. So I would like to know which are the main differences between logistic regression run through the machine learning approach and the traditional logistic regression.
Dear esteemed opponent, thank you so much for your compliment, and thank you so much for your comments. Regarding the logistic regression, and this is one of, I think this is one of the debate that is continuously occurring between epidemiologists, among epidemiologists, I mean, and among also machine learning professionals regarding when to say this is a traditional epidemiology approach and when to say this is a machine learning approach. Of course, there are certain algorithms that's obviously categorized as a machine learning, like, for example, neural network, which is definitely a machine learning or deep learning approach. But the cutoff point, I would say, or the threshold that we're saying here, we are moving into the machine learning is not agreed upon, and it's not clear. So we can, I mean, one of the main algorithm in machine learning approach is actually the regression, linear regression and logistic regression, which is already have been used for decades in the epidemiological studies. So I guess logistic regressions can be considered either less or less. However, I mean, it constitutes a main or a major algorithm that usually and commonly used in the machine learning approach.
Yes, because probably there is a difference in the selection of variables. So, and I think that the machine learning approach can be very useful when you have to analyze not only very huge data set, but you have a very large number of variables. So, in order to select these variables. While in the traditional approach, maybe the selection of variables or is decided a priori which variables include into the models, or you have other methods. But when you were discussing the results from the machine learning approach, you said that your results were comparable to those found in the literature in other studies. But I was wondering also if you, within your data, you run a logistic regression using the machine learning approach, and also you try to apply a traditional logistic regression, let's say with a priori decided nutrients, or if you did this comparison and you find difference in the selection of variables associated with bladder cancer and also in the quantification of the relative risk.
Thank you so much. Thank you so much, dear esteemed opponent. Regarding the analysis of nutrients in chapter 6, we conducted the algorithm for machine learning, but we did not do a traditional analysis. We compared the analysis or the results from our analysis from machine learning algorithm with previous studies from the same blend data set for some factors that have been already studied from some nutrient that have been already studied in the blend study. So we compared our results with the results of the previous traditional epidemiological, traditional analysis approach. However, we didn't specifically go to the step of analyzing, but this was one of the recommendations on the paper, that further analysis that look into specific factors, especially those factors, 3 or 4 factors that's most important, that have been identified in the machine learning approach, to be studied in depth utilizing the traditional analysis.
So I have another question regarding chapter 4 on the risk factor, on the association between soft drinks and asthma occurrence. So I would like to know more about the biological mechanism you already mentioned in your presentation, but you can say something more. And if you think you mentioned sugar, the possible role of sugar, but I was wondering whether other sugar-rich foods have been associated to asthma, and what is already known from the literature.
Thank you so much, dear esteemed opponent. Regarding the proposed or the potential mechanism of the association between soft drinks and asthma, of course, there are multiple hypotheses or theories regarding how soft drinks can affect the risk of asthma. The sugar content is one of the proposed mechanism. However, the current literature is not consistent, I would say, regarding the is not consistent to prove or disprove the role of the sugar in the development of asthma. So some studies shows that high sugar are associated with asthma, but one of the studies included in the in our meta-analysis actually compared the soft drink sugar sweetened with soft drink artificially sweetened. And in that comparison, actually, they found the increased risk among the artificially sweetened soft drinks rather than the sugar sweetened soft drinks. So this is a bit of conflict here between the studies regarding the role of sugar in the development. So this is make some authors suggest or propose that probably not the sugar itself, but the content or distribution of the content of the sugar. So focusing more on the, for example, fructose. So for example, in high fructose syrup, sweeteners that is commonly used in the soft drinks, although it contains 55% fructose, which is almost similar to the normal content of fructose in the regular sugar. But some studies found out that the fructose content in the soft drink reaches up to 65%. So it's a little bit higher. So this is also another mechanism that could have played a role in the association, in addition, of course, to the role of additives or the indirect effect of overweight and obesity.
Dr. Bossetti, I'm sorry, but
okay,
time's already up. That's also when you're having fun.
Okay, it's enough for me. Thank you very much.
Thank you very much. And we continue with the last opponent, Dr. Burza. He is Chief Health and Innovation Officer at the Health and Harmony Institute in Portland, USA. The floor is yours.
Assalamu alaikum, Abdulmohsen. Congratulations on your excellent thesis and so far your defense. My questions, I have a couple of questions to run past you, if that's okay. I think, firstly, an observation that in terms of methodologies, I'd like to know how you consider that the MMAs dealt with overlapping exposures. We know that in such ecological studies, this is very challenging, but just how well did you feel that this stratification was dealt with? And in particular, how did the studies and the analysis deal with the residual compounding effects of smoking and, to some extent, obesity, which are both major risk factors for both bladder and cancers?
Dear esteemed opponent, thank you so much for your compliment and for your comments. So regarding the MMAs and the overlapping problem, which is a challenge in conducting MMAs for meta-analysis, where the problem of combining more than one meta-analysis entail that probably we will have some studies in or shared between the two meta-analyses. So when combining them, we are running the risk of double counting and spuriously increasing the precision of the estimate, because we are combining the data from the same meta-analysis. So this is why we try to reach a compromise between not doing a meta-analysis at all, or combining the meta-analysis with a lot of shared studies and overlapping. So we set the rule to combine the meta-analysis only if the shared or the shared studies are less than 50% from the number of the smaller studies. So we have two meta-analysis. We look at the smaller meta-analysis. For example, it contains 10 studies. If more than 5 studies are shared with the other meta-analysis, we didn't combine the estimate. And we think that this is the best compromise that we say to get the overall estimate, but at the same time to avoid the overlapping or the double counting.
Okay. A second question, it was specifically to chapter 2, regarding the mechanism of selenium. Although higher body levels appear to have the strongest protective effect against bladder cancer, subsequent RCTs have shown no effect with regards to actual supplementation. And similarly with vitamin E, subsequent RCTs have actually shown that vitamin E supplementation increases the risk of bladder cancer relapse. So I'd like to know what your thoughts are on the mechanisms of this, and also how this realistically can impact policy recommendations.
Thank you so much. So yes, I agree that this is a challenge in the results of the studies, where randomized controlled trial experimental studies showed a different conclusion than the observational studies, and how and at that time it comes the question whether to trust the experimental study results or to trust the observational studies, especially if we have a large observational studies with well conducted. Because in experimental studies, we have the very controlled environment, very controlled consumption or dose and setting for the participants. So this is an advantage. However, in the observational studies, we have the real life situation, the real world situation, where these nutrients are consumed in the daily life, where this is what actually will happen for people. So various actually debates trying to explain why this is the differences. For example, that in clinical trials, we are singling out one nutrients and give them, while in the observational studies, the consumption is associated with other nutrients, other factors that may interact with the effect of the selenium, increasing or decreasing the effect of vitamin E, for example. So I think still say the jury is out about whether to trust the which evidence is more trustable in devising the population health advice.
Okay, but I'm going to push you a little bit more on that, though. I want, if you, as you now as a postdoc will be pushing policy and pushing research priorities, how do you see that in terms of your analysis of these well-conducted retrospective observational studies vis-a-vis the RCTs? If you had to take an approach in terms of a recommendation, what would you say in terms of this sort of output?
Yeah, thank you so much. Yeah, I think it needs further steps to analyze the available evidence So go into depth about comparing these studies and from experimental studies versus observational studies, utilizing tools that grade the significance or the strength of evidence to find out which evidence is more strong or stronger than the other evidence. I think more detailed analysis of these studies are required to come out with a conclusion. The strength of evidence or the grade of evidence is the best available tool for us in the recent time to find out or to compare conflicting evidence between various studies, whether from the same study design or from different study designs.
Okay. I mean, look, the thesis reveals a wealth of information in terms of risk factors. Oh, was that the end?
That is the end. Yeah.
Okay, I will leave it there. Thank you so much.
Thank you so much.
Yeah, okay. Dear candidate, as you probably have guessed, the time for your defense has passed. And the degree committee now will go to a breakout room to discuss your thesis and the quality of the defense. And I kindly request you to wait until we come back to you.
Thank you so much.
Dear Abdulmohsen Alzalabani, the degree committee here, present online, has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Zegers is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor. Professor Zegers.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Promise.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Abdulmohsen Hamdan Alzalabani, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, now shown by the beadle.
Thank you so much.
And I give now the word to Dr. Mercedes.
Dear Dr. Abdulmohsen, today is your special day. And even though I can't congratulate you in person, I'm honored to celebrate your success virtually. Doing a PhD is never easy. It's full of challenges and discoveries, but you handle it with a great effort, creativity, and strength. Your research on diet, lifestyle, and health has been impressive and practical. You've shared important findings, such as how changing lifestyle and reducing work-related risks can lower the chances of prostate and bladder cancer, how drinking tea may reduce the risk of bladder cancer, and how reducing soft drink intake can decrease your asthma risk. To me, these insights go beyond academic value, because they might provide real-world solutions to everyday health challenges. What made your work even more special were the difficult methods that you used. You're already a skilled epidemiologist, but learning about machine learning was a new and tough challenge. And I was really impressed by the way you took it on with patience and persistence, and how you managed to use these advanced tools to answer your research question. This showed not only your hard work, but also your remarkable determination and personal strength in handling such a challenging topic. Your proposition about epidemiological teamwork particularly interested me because of our own collaboration, which at least to me is a perfect example of it. And of course, I hope you agree. Our journey began in a rather modern and unexpected way on Twitter. I still remember your reaction on the advertisement of our group. Little did we know that a simple online interaction would lead to such a meaningful and unique research journey. From that first Twitter exchange to countless Zoom calls, we built a partnership that was truly special. We only met once in person, but Zoom became our virtual workspace, where we connected, problem solved, and grew as colleagues. Despite the physical distance, you always brought focus, dedication, and a good atmosphere to our meetings, making it a real pleasure to work with you. To be honest, supervising you didn't really feel like supervising at all. It was more just great teamwork. You worked very hard, taught me a lot along the way, and I can only hope you gained some insights from me as well. I think together we tackled the challenges of research and learned from every step we took. Looking ahead, I truly hope we can continue this collaboration in the future. As we discussed, the idea of supervising new PhD students together is an exciting one. I have no doubts that if we move forward with this idea, it will lead to many more shared successes, discoveries, and valuable learning experiences. I really hope this will happen. Once again, congratulations on this incredible milestone, I'm looking forward to what the future holds for us. Thanks a lot.
Thank you so much. I appreciate it.
Dear Dr. Alzalabani, dear young Dr. Alzalabani, by tradition now the first 6 weeks we call you the young doctor, so and you already know. Also on behalf of Maastricht University, I congratulate you with the degree you have acquired. We are very proud what you did and how you did it. We are very happy to have you as an alumnus of our university. Since this is an online session, normally you would go to a big stair and have a picture taken there. But now you have the recording of the session, which works almost as well. And I would like to give the other people the opportunity of congratulating you online as well. So I first go to a very proud Professor Zegers, who probably wants to congratulate you.
Congratulations. We just had the meeting in the committee, and everybody was impressed about both your presentation and your defense. So I can only echo what Anke said. The collaboration with you was always smooth. You worked always really hard, and we hope indeed that we continue our collaboration. This is just the first stepping stone of another decade of collaborations.
Thank you so much.
Well done, Abdulmohsen.
Thank you so much. Appreciate it.
Dr. Veselius?
Congratulations again.
Yeah.
I think I mentioned what I wanted to tell you. So yeah.
Okay.
Let's meet soon again.
Yeah. Well, normally we would stand in line and have handshakes, right? So, Professor Smith.
Yeah, congratulations, Abdulmohsen, with this degree, and also congratulations to your promotion team and your loved ones. And I wish you very much success and happiness in your career and further life. So you did a good job. Cheers.
Thank you so much. Appreciate it. Thank you so much.
Professor Jonkers.
Yeah, also for me, congratulations with a well-deserved doctor title, also on behalf of the Research Institute. I think it's a very nice thesis, very nice defense. I enjoyed it a lot, and I would really like to follow up with your supervisory team to see how I can learn from that and use it, and would be really nice to see you in academia in the near future. So good luck with your further career.
Thank you so much, Professor.
Professor Selim.
Congratulations, Dr. Abdulmohsen, for this marvelous thesis, and congratulations for the supervisor team, Dr. Ziegler and Dr. Wislis. Hoping continuous advanced and sophisticated research in the future. Congratulations
Absolutely.
again.
Thank you so much, Professor.
Dr. Borsetti.
Dear doctor, congratulations also from my side. So I understand from your supervisor that you did a great job, as we saw in your presentation, and they were very satisfied, and also there was a very good collaboration with you, and this is very nice. So I wish you the best for your professional life, and congratulations again.
Thank you so much. Appreciate it, doctors.
And finally, Dr. Burza.
Dr. Abdulmohsen, you have to get used to hearing that name now. Congratulations on the thesis. That was wonderful to read, very interesting, and a very stimulating discussion. And I wish you only the best for your future academic and professional career. Congratulations.
Thank you so much. I appreciate it, Doctor.
Okay, thank you everyone for participating, and I will now officially close this session. Have a very nice day all, and I'm going for a coffee because Dr. Selim has the coffee machine already behind him, so